and save ourselves from these zoonotic disorder. Thank you, Doctor. Welcome.
Well, thank you very much for that introduction. I'm not sure I can't live up to the billing. That's rather a tall order you've put to my feet. Thank you also to the organizers of this, and particularly Dr. Zobta Shinwari for directly inviting me. It's always great to follow Nancy and David, and very good to hear David talking about leadership in very much the same ways as I think about it, and the need to build expertise before events happen. Expertise being a sort of, in the way that is helpful in these situations, is that level of knowledge about a subject where we can instinctively think about problems rather than just have book knowledge. So great to follow on to that. Why I'm talking about this tonight is that, or this morning in your time, is that, just put this onto, I don't know if that's worked.
Yes, sorry. Now it's better. Thank you.
Right, thank you. So why am I talking about this subject? Why am I talking about how to stop something which doesn't exist yet? I've had the honor to work with Dr. Amar Ikram and the National Institutes of Health of Pakistan over the past year and a bit, trying to develop a national infection prevention and control plan for CCHF, which of course is a well-known zoonotic disease. And that was a wonderful experience, but it got me thinking, particularly when Dr. Zobta Shinwari asked me to come up with a talk for this meeting about what I've been doing in the past 10 or so years. And within the past 10 years, I've worked in Nigeria on Ebola. I've worked with refugee camps and with institutions in Pakistan on COVID. And last year, I've been working on CCHF. All three of these are zoonotic diseases. And so it's raised the question, is there anything we can be doing now to slow down the emergence of new viral zoonotic diseases? A long time ago, I read Jared Diamond's book on guns, steel, and germs, or whatever it was called. And in it, he posited the idea that as the world gets more crowded, so the types of diseases that we're going to be exposed to will change. So it's gone from single cellular organisms like parasites down to bacteria, then to viruses, and now we're getting exposed to prions. But this idea that the population density is something which is very intimately connected to the types of disease we're going to be exposed to is something that stuck with me. And it's particularly stuck with me in my thinking about how do new zoonotic diseases arise? How is it that these viruses are jumping from animals to humans? And if you think about it from the fundamentals of the science, zoonosis is essentially evolution in action. And evolution requires replication, it requires reproduction, so that you can get the assortment of genetic material, and it requires selective pressure. So if zoonosis is evolution in action, it needs passage through human cells to have both that replication and the selection pressure. So how then do animal viruses get to infect and become adapted to human cells? Well, if we think in terms of what it takes for a virus to replicate, they need to attach to a cell. They need to penetrate the cell. They then need to release their material, their genetic material into the cell, hijack the cell's machinery for genetic material reproduction and for protein production, assemble the proteins and the nucleic material into the new variants and then release it. But if we think about that in terms of zoonosis, what is the most important first step of getting those animal viruses into human cells so that selective pressure for evolution can happen? And it really is, sorry, something's going wrong with the slides here. Okay, it really is this first step of attachment and penetration into the cell, which allows infection. There's no evolution without selective pressure. There's no selective pressure without replication, but there's no replication without infection. So really, I think this is the first step of the process of an animal virus becoming able to adapt to becoming a human virus, is that it has to be able to infect a human cell. So how can that happen? Well, there's basically two routes that you can have that first infection from an animal virus into a human cell. Through spillover, which Dr. Zabtashenwari has talked about already, and through viral propagation of animal viruses in human cell lines by scientists in laboratories. Looking at the probability of spillover, essentially the probability is determined by three types of factors: disease dynamics in the reservoir hosts, the degree of pathogen exposure that humans have, and then between humans interactions and humans' susceptibility to infection of the disease in the first place. There's a very good slide from this paper by Plowright and company in Nature Review of Microbiology, where they talk about these three levels of, at the top we have the basic pathogen load in the reservoir, and then we've got the potential exposures routes through excretion of the virus from the reservoirs, or through the slaughter of the animals by humans, or vector-borne through insects or ticks or fleas, that sort of thing. And then at the bottom we have the human susceptibility to infection. So these three functional phases are the pathogen pressure, the amount of pathogen available to expose to human hosts at a particular point in space of time, human vector behavior, and then our human susceptibility to infection. Sorry, I'm having problems advancing slides here. If we sort of ally this then with normal thinking about infection prevention and control, and how that then might relate to a newly emerging disease. When we talk about infection prevention and control, we think about the chain of infection. We think about the 6 factors. What are the characteristics of pathogen? Where is the pathogen found? What are the reservoirs for the pathogen? How do they get to infect us? How do they get between hosts? That is, how are they transmitted? How do they get out of host once a host has been infected and go on to infect someone else? And who are the vulnerable people? So we can think about newly emerging zoonotic diseases and how to control them in terms of those 6 factors. But if we don't know, if it's not yet with us, then we can't really characterize the pathogen, but we can start thinking about where are the likely reservoirs and what are the likely routes of infection and transmission. Again, in terms of general infection prevention and control, we think in terms of mode of infection as being either general transmission through abiotic environmental factors like wind and water, or through animal vectors, be they rodents or insects. And then we've got human-to-human transmission. But at that first instance, we're really talking about this general transmission. How is it going to get from an animal reservoir to a human in the first place, to make that first infection so the process of evolution can start happening to create the zoonotic disease. And again, in terms of infection prevention and control, we think about portals of entry and exit. How can the virus get into the human body? And once the human body's been infected and reproduce the virus, how does the virus then get out of the human body to go on to infect someone else? And I think once you put all these together, that gives us a conceptual framework for thinking about how we might create barriers to spillover of an as yet to evolve zoonotic disease. We want to understand that there are these relationships, and Dr. Zubda Shinwari talked about these quite eloquently. There's relationships between animals, humans and their pets in the where we have frequent contact with those types of animals. There's frequent contact between people involved in farming and livestock, or people involved in the processing of animal products like meat or pelts or skins or wool. Those humans involved in those professions have regular contacts with those animals. So what sort of effective barriers can we put up to prevent infection between those animals we come in contact with most frequently? Physical barriers, behavioral barriers, good process barriers. What are the barriers then that we need to put up around all of us, the animals that we have frequent contact with and ourselves? And the other big issues out there, like the vectors, like the pests, like viruses that might be in the soil, air, and water. And particularly, how do we protect ourselves from contact with from those ecosystems where these novel viruses are likely to evolve or get to the stage where they go from a virus that cannot infect a human to one that can infect a human, even if it then is not able to be transmitted human to human. But how does it evolve? Where is it evolving to the point where it might create that first infection through spillover? And once we start thinking about it in these sort of conceptual ways, what are those barriers that might be effective, both in terms of behaviors and in terms of physical barriers? And these are really early thoughts, so I certainly can't live up to the request that you posed to me, Doctor, at the beginning. These are just some very early unformed thoughts. We certainly can start thinking about how do we minimize contacts between humans and the animals that are regularly in contact with humans, like our pets and our livestock. With those incubator environments, if you like to call them that, those areas where the new zoonotic diseases are most likely to be fermented so that they can actually effectively spill over from being a purely animal virus to one that can cause infections by direct contact between the animal and the human. And then how do we go about controlling pests and known vectors that are likely to be future vectors as well, so that they're less likely to be the route by which that first infection is made? What sort of barriers are feasible to put up between wildlife habitats, which might be these incubators for the new infections, and the livestock that we have? How do we create those barriers between livestock and wildlife? How do we protect species diversity within wildlife habitats? Because there's fairly strong evidence that the narrower the number of species are within an ecosystem, the faster virus evolution happens within that ecosystem based on less species diversity. What sort of good pasture grazing and management practices can we come up with to, again, protect our livestock from getting infected and becoming intermediary species between the wildlife reservoir and us. And likewise, disease surveillance for that livestock. How do we limit, again, as Dr. Zobda Shinwari said, how do we go about limiting the use of wildlife as a protein source when so many in the world depend upon it? The last time I looked at this, I think in Africa alone, 1 million pounds of wildlife was used for protein per year by the population in Africa. So if people are dependent upon this as a protein source, what do we need to do to make it so they don't have to depend on this as a protein source? Do we need to limit virus hunting and collecting, because that is actually bringing in viruses from these potentially incubator environments into human laboratories and into contact with humans? Is that a source of risk? Do we need to have a discussion about that? Do we actually create huge risks by propagating non-human animal viruses in human cell lines? Is that something we need to discuss about? How do we, about reducing that as a practice? And how do we go about getting a global disease surveillance program together, looking for unknown diseases so that we can spot emerging zoonotic diseases at a very early stage before they become well adapted to the human model and have very fast, effective human-to-human transmission. Take, for example, as you said, the monkeypox at the moment. At the moment, transmission human to human is very inefficient. It requires prolonged close contact between people for that transmission to happen. But the fact that it is transmitting from human to human means that it now has those two factors available for evolution to be a closer fit. It has reproduction in human systems, and it has a selective pressure to actually transmit more effectively. And so the sooner we can stamp out the outbreak of monkeypox, the less time we give the virus to better adapt to human transmission, and the more likely we are to prevent it becoming a much more transmissible disease, which is a greater threat to humanity. So that's my basic early thoughts. These are very early thoughts on it, because this is a subject which I know many people are thinking about and many people are struggling with because of the complexity of the issues involved. I think we do have some areas of research that we might want to look at, like what factors do speed up viral evolution in wildlife habitats, in those incubator environments where the new viruses might emerge that are capable of making that at first infection. Like which species are most likely to be the sources of those future zoonoses? So is it pigs because of the similarity of their serology to humans, or is it birds and bats because they maintain much higher body temperatures, and therefore the viruses that evolve in them survive human fever as a response to infection, and therefore are more likely to cause diseases in humans if they infect in humans. So what are the implications that research might provide for how we go about minimizing future threats? Is it important to know the locations of wildlife habitats that might be especially good incubators for spillover diseases and for known vectors, and where they are in the world in relation to livestock? So those are just some ideas for how we might go about it, and a framework for thinking about it, and some research ideas. But as I say, this is very early thinking for me at this stage, and I've enjoyed the opportunity to offer these ideas to this forum, and very much look forward to feedback and help in trying to develop these ideas further. Thank you very much.